Immunology Insights, Basics and Beyond, Chapter 21, Clonal Selection and Expansion. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our last chapter, we explored antigen presentation, a critical process where immune cells display antigens to activate T cells. But once a T cell recognizes an antigen, what happens next? How does a single activated T cell lead to an army of immune defenders? That's where clonal selection and expansion come into play. Today, we will discuss how the immune system amplifies its response by selectively expanding antigen-specific lymphocytes. This mechanism ensures that when a pathogen invades, our immune system can respond swiftly and effectively. So let's talk science. Let's start with what is clonal selection and expansion? Clonal selection and expansion are key processes in adaptive immunity, enabling the immune system to mount a targeted and robust defense against pathogens. The concept was first proposed by Sir Frank McFarlane Burnett in the 1950s as part of the clonal selection theory, which explains how immune specificity and memory arise. Let's have a closer look. First at the clonal selection. When an antigen-presenting cell or APC displays an antigen, only T or B cells with receptors specific to that antigen get activated. Second, we have then the clonal expansion. Once activated, the selected lymphocytes proliferate rapidly, generating a large population of identical cells that can fight the infection. This process ensures that only the immune cells with the correct specificity are amplified, preventing unnecessary immune activation. Let's have a look at the role of T and B cells in that context. First, the T cells. After activation, T cells undergo clonal expansion and differentiate into effector T cells, so CD4 positive helper T cells and CD8 positive cytotoxic T cells that coordinate and execute the immune response, or memory T cells, which persist long term and provide faster responses upon reinfection. And second, the B cells. Activated B cells differentiate into plasma cells, which secrete large amounts of antibodies, or memory B cells, ensuring rapid antibody production during future encounters with the same pathogen. Now let's see how clonal expansion actually works. First, we have the antigen recognition. A naive T or B cell recognizes a specific antigen presented by an APC. Second, we have activation. The immune cell receives co-stimulatory signals to ensure proper activation. Third, we have proliferation. The selected lymphocyte undergoes multiple rounds of cell division, creating a large amount of clones or identical cells. And fourth, we have the differentiation. The newly formed cells specialized into effector and memory cells, equipping the immune system for an immediate response and long-term immunity. Of course, clonal selection has a high impact in health and disease. Clonal selection is vital for the immune defense, for example, in the vaccine-induced immunity. Vaccines work by triggering clonal selection and expansion of memory cells, providing long-lasting protection against infectious diseases. However, dysregulation of clonal selection and expansion can lead to several diseases. Autoimmune diseases, for example. If self-reactive lymphocytes are mistakenly activated, they can attack the body's own tissues, for example, in multiple sclerosis or type 1 diabetes. And cancer, of course. In some cases, abnormal clonal expansion of immune cells can lead to conditions like leukemia and lymphoma. To conclude, clonal selection and expansion allow our immune system to generate highly specific responses and establish long-term immunity. Without these processes, the adaptive immune response would be slow and ineffective. In our next chapter, we will explore the memory response, which ensures that our immune system remembers past infections and can respond more efficiently in the future. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So see you next time and thank you for listening.